By the end of this presentation, I'm hoping that you will learn or that I deliver three messages. The first one is going to be what is or what are lichens, what is the web of life, and why we should care about it, and what do lichens have to teach us about that web of life. And so I'm gonna try to be delivering this message through my personal experiences and not knowing and getting to know and starting to love lichens, becoming very passionate, and also through some of the research that I do here at Duke as a PhD student. So I'm gonna start here, um, which is where I wish I was right now. So this beach is 20 minutes away from my parents' house in my hometown where I grew up, which is right here in the Caribbean coast in the northern part um, of Colombia. And so the reason I'm showing this to you is because I want you to know how um, for a very long time I didn't know what lichens were for most of my life, hadn't even heard the word. So this is where I grew up. This is the landscape that I was surrounded by, and this is a very hot place. And it's characterized this ecosystem by having very long periods of no rainfall, so it's very dry. This picture here, I took it probably a couple of days after it rains, but most of the time it looks like this. This is from a similar angle. So this is a seasonal dry forest. So it gets so dry for so long that most of the plants that are around here drop their leaves for a long part of the year. And so the thing about this is that in this ecosystem there are lichens, but they are not really conspicuous. So I grew up without ever hearing of them or seeing them. So just so you get a sense, I borrowed these pictures from a couple of studies that have described the lichen flora from that area in Colombia recently. And at this point, I still haven't told you what lichens are. I didn't know either. But just so you see that the colors, this is what they would look like if I saw them. Um, the colors are not very, in most of them are not very conspicuous, and they are growing in a surface. They're not really coming out, just kind of creeping in. And if you look at these scale bars, they are 1 millimeter, so they are also very small. So unfortunately, they are all very interesting, but I never paid attention to them, never even knew they were there. So for my, when I went to college, I moved from the um, Caribbean coast to Bogota, which is the capital of the country. And the city is very high up in the Andes Mountains, like 2,600 meters above sea level. And so the landscape is very, very different. So there I was in Bogota, my first year in college, first class as a microbiology student, organismal biology. And we took a field trip to this to a national park that has a lot of this ecosystem called páramos, which some people also known as tropical alpine. And so the word páramos, páramo in Spanish, literally means soaking wet. So this place was the complete opposite of the landscape in the ecosystem where I had grown in. The vegetation was very different. The feeling of it, as you can see by the pictures, was very different. There's a lot of wetlands, hyper diverse. And I was fascinated by it. Among other reasons, because in these ecosystems, lichens were a very conspicuous part of the landscape. So I'm borrowing these pictures from a field guide by Robert Lukin and Viviana Moncana, two very prolific lichenologists that have described a lot of the flora in Colombia. And as you can see, the colors and the shapes that the lichens take in this place are very different to what I showed, to what are to the ones that are more common in this dry forest. So they are coming out, they're more colorful. A lot of them form displays like this. So again, I still haven't told you what lichens are at this point, but when I started going into these places, I was fascinated because I was seeing all the shapes, and there you cannot miss them. There's just like in your face. So it was at that point where I became interested in actually learning um, what a lichen is. And so a lichen is essentially a symbiosis, which means that you have uh, organisms that are living together. So in this symbiosis, and we're gonna be um, learning some terms here. So in this symbiosis, you have a fungus, which lichenologists call a mycobiont, that lives together with a photobiont, which is going to be an organism that can do photosynthesis. So when that fungus is living with a green alga, we call that they form what we call a chlorolichen. Sometimes the fungus can be with a cyanobacteria, which is a kind of bacteria that can do photosynthesis. And in this case, we call them cyanolichens. And sometimes some lichen forming fungi can be in symbiosis with a green alga and a cyanobacteria at the same time. So we call those H-remember lichen, like the one that I'm showing here. You're gonna be seeing a picture of this particular lichen a lot because it's one of my favorites. Um, and so this very bright, beautiful green color is given by the green algae, and then the cyanobacteria, in this case, are embedded in these darker uh, spots that you can see on the surface of this. So lichens are what biologists, the symbiosis is what biologists call a nutritional symbiosis. So they are living together and they are exchanging nutrients. So the photobiont, as I said before, is able to photosynthesize. So it provides the fungus with carbon in the form of sugar, which it uses for energy. And when the photobiont is a cyanobacteria, it can do photosynthesis, but it can also fix nitrogen directly from the atmosphere, which organisms need and the photobiont takes. And we don't know as much about what flows in the other direction, so what the fungus is giving back to the photobiont, but we know that there is some other micronutrients at play. We also know that the photobionts, so the green alga and the cyanobacteria, they can be free living. You can find them outside of the lichen. Whereas for the mycobiont, the fungus, we don't have, most of the evidence points to them not being able to survive on their own. They need the photobiont and be as a lichen to be able to exist. So in this scheme that I'm showing you here on this diagram that I'm showing you here on the left, you have what the fungus looks like when it's growing on its own in this petri dish. And this is what the alga, a green alga in this case, looks like when it's growing on its own. And then when they come together, they form this structure that is very distinct to what each of them looks like when they are growing separately. So it's kind of like a characteristic of this symbiosis that they form this very distinct unit when they come together. There are about 20,000 described species of lichen-forming fungi and about 100 species of different photobionts, although these numbers are growing very quickly, and we'll talk about that later. And an important point is that lichenologists refer to the lichen as a whole using the name of the fungus. So for example, this one here is from the genus Cladonia, and Cladonia is the name of the fungus in this case. So a little bit more about this unit that these two things form when they come together, which we call a thallus, the lichen individual, is that so if we took a section of most lichens, we would find that they are stratified. You start seeing some layers that are composed of the two photobionts in different parts. So most of them would have a cortex, as shown here, that is mostly, or that is the mycobiont cells, the fungus. Then there would be another layer where you have both cells of the photobiont and filaments of the mycobiont. Then you have another layer, which we call medulla, which is just the photobiont. And then many of them also have another lower cortex, which is also just the mycobiont. Now, recently, and maybe you can imagine from the structure, there's been a lot of findings that these structures, the thallus, can harbor a lot of diversity beyond the main partners, the photobiont and the mycobiont. There is tons of bacteria and even other fungi living in many within this the thallus, the thalli of many of these lichens. And that has led some people to even want to redefine the lichen symbiosis as a complex ecosystem because of all of these parts. But we're gonna keep it simple and think about lichens, about this symbiosis, so this fungus and this photosynthetic organism living together. One of the more interesting and important aspects of the biology of the lichens is that they can dry out and get wet very, very quickly. And this is super important for their success in nature. And that also makes them look very different when they are dry versus when they are wet. So again, this is the one that I told you you're gonna be seeing pictures of, my favorite lichen. Look at what it looks like when it's dry. It looks like nothing, but then you add some water to it and boom, it comes to life. And so this is, by the way, one of the reasons why when I started going to these paramos, I could not miss the lichens because it's often so wet that they are in full color. And so this transition between dry and wet happens very, very quickly. So I have this video here to show you, and you have focus, this is the same lichen. Focus here in this part that is kind of grayish. It's dry, and so I started adding water to it, and you can see how the color is changing very quickly as this thallus is absorbing the water. By the way, I know this is not the most glamorous way of adding water to it. So if there are any lichenologists or bryologists, often we would bring a spray bottle to do this in a more elegant and gentle way, but I didn't have mine that day. So I was just throwing handfuls of water at it. But anyway, the point is that they can take up that water very quickly, and you saw how it changes the color. And they can also dry very fast. And that's very important because the water, they absorb the water, they need the water so that the photobiont starts doing photosynthesis and start producing food for the whole thallus. But then when water is no longer super available in the environment, they dry out very quickly and they kind of lower down their metabolism. And that is a strategy to keep some other stuff from growing in them, like mold, for example. So if you go out and you collect a lichen and you put it in a Ziploc bag so it doesn't dry out, you would see that its mold starts growing on it very quickly. So this is kind of one of the strategies that they have. So by now we have a kind of an idea of what a lichen is the symbiosis, fungus, some photosynthetic organism. We know something about their diversity and how they live in nature. So let's go now into the second point that I mentioned, which is what is the web of life and why we should care about it. So the web of life is, I'm referring to the idea that biodiversity is not just a collection of species in nature. Those species are always interacting with one another, and they effectively, the collection of those interactions forms a web of interactions. And so to build up to why we need to think about it that way, why we should care about it, we're gonna try to break it down to the basic of what an interaction is. So what I'm gonna do now is I'm gonna show you some pictures of some different kinds of interactions. And in the meantime, you try to start trying to develop a definition of what an interaction is. So for example, I have here this mosquito biting this human host, taking its blood, potentially transmitting some parasite, I don't know. This picture here is another interaction. These are plant roots in green, and then these spheres that are around it are cells of fungi that colonize it, called mycorrhizal fungi. And then they form another symbiosis where there's also an exchange of nutrients. We have here our now familiar stratified section of a lichen with the fungus and the photobiont interacting. Here's a picture of this moth pollinating this flower, getting food while the plant gets help with its reproduction. Um, this is a um, microscopic picture of some bacteria that are found in our gut. And this one of my favorites. And by the way, one weird thing about me, I really enjoy uh, seeing predators devour their prey. So I was very happy when 2 days ago I saw this Cooper Talk. Um, right, I caught it in the moment where it was stripping the guts out of this poor squirrel to feed. So anyway, we have here, different types of interactions. They're all very distinct in nature. So what is an interaction? How can we come up with a definition that encompasses all of these different things that are so different? So if you think it seems like a trivial thing, you could recognize that there is an interaction, but it's difficult to come up with a definition. So we are going to use what ecologists call a phenomenological definition, which is just a long way of saying that it's kind of a lazy definition. It's gonna tell us something, but not a lot. But in this case, it's gonna be enough. So I'm gonna say that two species are interacting if they affect each other's population sizes. So if there's changes in how many are there of one species, and that change causes another species to change its population size, then we're gonna say that they are interacting. And that doesn't have to go both ways. It can just go in one way or another. If you start thinking about these examples that I gave you, one way or another, they all result in changes in the sizes, either by promoting reproduction or providing food, etc. So now that we know we have that definition of an interaction, we can build from pairwise interactions into webs to get at this idea of why thinking of these webs is important. So I'm going to introduce an idea that is very important to understand how ecologists study these interaction webs, which is the fact that you can represent them as a matrix. So in this cover picture that I had this very nice cartoon of an actual interaction web. And so what we're gonna do now is we're gonna build that web in parallel with an interaction matrix so that you get a sense of what it looks like. Because the thing is that these web illustrations, they look very nice, but we cannot extract a lot of information from it. So just so you get the ideas, we're gonna do this exercise. So let's say that I'm studying this thing, which is a representation of a cyanolichen. And in this matrix, I'm going to have lichens, mycobionts in the columns, and photobionts in the rows. In this case, the photobionts are going to be cyanobacteria. So I have this lichen mycobiont, and then I collected and I studied, and I found that it's associated with this cyanobacteria. So boom, I have here, they are connected by this link, they form this interaction. Now I add another lichen, this is another cyanolichen, I look, oh, it's interacting also with this cyanobacteria, it gets connected here. Find this other one, which is again my favorite one, it's interacting with this cyanobacteria, bam, it gets connected. Now, this one, I found another one that is now connected to this other cyanobacteria, and so forth, and so forth, and so forth. So here, what we did, basically, we built this web representation as a matrix. Now, if we look at this cartoon here, it should start becoming evident why thinking about these webs is important if you remember how we define an interaction, these changes in population sizes. So let's say that something, for an external reason, something happens to how many of these lichens there are in a particular place. Since these two things are connected through this interaction, that change in the population of this is going to cause a change in the population of this. But this guy is also connected to this and that. So a change here might cause a change here and here. And now this one is connected to this, and so you can see how something that starts here in terms of changes to the population can end up affecting this guy that is way over here, even though they might not be connected directly. And this is actually how biodiversity functions. And population sizes, whether they go up or down, it determines whether species are going to go extinct or not. So thinking about biodiversity as a web is important because that is how they actually function in terms of how changes spread through through these interaction webs. Now, this one of the reasons we do this with a matrix, as I said, is because it's difficult to extract information from here. But it turns out that when we have this representation, and because of some convoluted combination of differential calculus and linear algebra, we can actually quantify, based on the structure of this web, whether the species that are part of it are going to persist in time or not, whether some are going to go extinct or not. So we say that the structure of these webs tells us something about the stability of the community. So that's why we think of this and study them, these communities, as webs. So how do we talk about the structure of them? So let's use this example to see something here. So what we built here, you can see that this particular schematic web has these three species, for example, that are all interacting with this cyanobacteria here. And then these other three that are interacting with this guy. So there's like two subsets of species. They're all connected, but there's two subsets that are preferentially interacting with one another. So when you have that kind of structural motif, and you can see it here, you call that module. So there's a module here, and there's another module here. So there's ways to quantify that. And again, that contains information as to whether or not these communities will persist in time or not. And if we want to manage biodiversity, this starts becoming important. Now, as I mentioned earlier, this particular type of interactions in lichen, both partners are benefiting from the interaction. So we call this mutualistic interaction, and we call these things mutualistic webs or networks. And so traditionally, the structure and the properties of those webs have been studied with these iconic mutualisms between plants and animals, such as the situations where birds, for example, eat the fruits that some plants produce, and then they disperse their seeds. So the bird is getting food, and doing that is helping the plants disperse. Or pollination systems where the animals are getting food from the plants and they are aiding the reproduction of the plants. So we have learned a lot from studying these interactions as networks, but there is a feature about them that makes them difficult for a particular kind of questions. So the thing about these interactions is that they are transient. The way people study them is that basically you have to go and sit somewhere and start counting how many times any particular bird comes and eats a fruit and disperses its seed, or document and count how many times some bee or some other insect visits a flower. But if you have looked around you in your backyard, that, for example, the flowers are not all blooming at the same time in a year. The flowers that you see in the early spring are not the same ones that you see in the early summer, and they're not all the same ones that you see in early fall, for example. And so, what that means for studying these interactions is that they are changing effectively very quickly at the scale of weeks or months. So, that makes it very difficult to document them over very large spatial scales, because if you have to go and sit and watch these things and you want to do this, say, over all North America, you start in March and you finish in August, but by the time you're sampling in August, the things are not the same as when you started back in March. So it makes it difficult to make comparisons because it's difficult to sample them over very large spatial scales and with a very fine grain. So that's the kind of questions where lichens are a very good model system to the ones that pertain how these webs are structured in space, because lichens form a persistent association. So when the microbiome and the photobiome come together, they form this distinct unit, the thallus, um, that has a much slower turnover in space, so you can sample over years without having to worry too much about those interactions changing during the time that you are sampling them. So that's why I use lichens as a model system to address precisely that. How are these webs structured in space? And how do they change when we look at ecosystems at different scales? So I do that research using this group of lichens from the genus Peltigera. So it's a group of cyanolichens. So it's the fungus is Peltigera, uh, and they associate with a cyanobacterium that is called nostoc. And this is what they look like. So again, my favorite green one is here. You'll keep seeing it. And this one is green because it's a tree-membered one. So some species can have both nostoc and another alga. And so one thing that is fundamental to characterize those webs is our ability to determine who it goes into these bubbles that I put in this representation. So this, as I said earlier, these bubbles are supposed to represent species. So basically, we need to know what the species are, and we need to have a way to recognize them in order to even start thinking about studying their interactions. So one of the reasons I studied this particular group of lichens, Peltigera, is because we know a lot about it, a lot about what the species are and how they're related to each other. So this group of lichens has been studied for over 20 years by people in the lab where I'm doing my PhD and outside. So here's Francois Luzzoni, who is my advisor, Yolamia Dikowska, who is a senior researcher at Duke, Nicola Maga, who has done a lot of work studying those questions, Trevor Gore, an expert in lichens in Peltigera in Canada. And so they've been collecting these lichens all over the world. And through a lot of studies, they have elucidated what are the species and how they are related to each other. So in this figure that I'm showing you right here, this is a phylogenetic tree, which is essentially a genealogy that shows how species are related to each other. And I don't want you to look at the details. This is just to show you that we know what the species are and how they are related, as shown in this genealogy, and that knowledge has come from looking at those species all over the world where they occur. So we know a lot about what the species are, how they are related for the fungal side of these lichens. Now, what about nostoc, which is this cyanobacteria that are part of this symbiosis? So the situation there is a little more complicated. So I have this, again, this is another genealogy that is showing, or phylogenetic tree that is showing the relationships between different types of nostoc that are here named with Roman numerals. And so what I want you to look at in here is that this tree has some branches that are thick and some that are not. And so the places where it's thick, it means that we are very confident about that particular relationship. So where it falls, how it's related to each other. And when it's not thick, it means that we're uncertain about it. And as you can see here and here and here and here and here, there is a lack of thick branches in a lot of these genealogies. So we have a lot of uncertainty about the relationship between these different nostoc. And a lot of it is coming from the fact that we build these genealogies by sequencing a small part of the genome of this cyanobacteria. And we have sequenced so much, so many different ones, that we have exceeded the amount of information that we can extract from it. So one of the things that I'm doing for my PhD is try to tackle this by using genomic data, full genomes, to see how these nostoc are related to each other. So I've been doing that, and actually we don't have this problem anymore. There's some preliminary data. So I have been sequencing dozens of genomes, and what I want you to look at here is that as opposed to the genealogy, the tree on the left, now we have a lot of thick branches in this tree. So it seems like a simple thing, but it matters a lot to know how these things are related to each other, because then we can start making sure that the units that we're keeping track of to characterize those interactions make sense. Now, we know how they're related, but what about species? So let's step back for a second and think what even is a species. So some of you may know that this is a contentious topic in biology. There is a myriad of definitions, but species are a fundamental unit of diversity. And there's one of the most common definitions has to do with whether or not two organisms are able to breed and produce fertile offspring. So I'm going to use this kind of classic simple example to illustrate it, which is we have a horse and a donkey. Sometimes horses and donkeys can mate and they can give mules as offspring, but the mules cannot give birth to other mules. They are sterile, essentially. So we say that horses and donkeys are part of a different species because they cannot give fertile offspring. Now, at the core of this definition is the fact that these organisms need to be able to have sex and reproduce sexually. But nostoc is a bacteria, and bacteria don't really have sex. The way they actually reproduce is that they essentially clone each other. So if you start with one nostoc that I'm showing here is this thread of cells, it will give rise to two nostoc clonally, and those two will give rise to two more, and so back and so forth. So if they don't have sex, how can we our species in this bacteria even a real unit in nature? It turns out that they are able to exchange genetic material even if it's not the same way as in sex. So people have for a long time been trying to find criteria that would help us delimit species in bacteria, which would be very useful for me to keep track of these interactions. So even though we are not certain that species are real in bacteria, people have been naming them based on other criteria anyway. And there was this study recently that took 90,000 genomes from a database that has genomes of many bacteria. And then they compared them all against each other, all of the pairwise comparisons. Okay, and they look at how similar each pair of genomes were. And they found something very interesting. Every time they compare something that they thought, two things that they thought were the same species, they were between 95 and 100% similar. But every time they compare two genomes that they thought were from a different species, they found that they were 82% or less similar. So essentially, there was a gap in the similarity. And when I saw this, I was very excited because I said, aha, I can use this as a criteria to delimit my species. And I'm going to have the genome, so I'm going to be able to do that. Turns out when I started sequencing this genome that I told you earlier, I have over 100 of them. I started doing the same, comparing them, and I found no such gap. So it turns out that in the cyanobacteria, they are kind of like in a continuum of divergence. So one of the things that I'm interested as part of my PhD is to try to find other criteria to delimit uh, really solid units to keep track of, and understand the processes that lead to their formation. But for now, the classification that we have is going to have to do. So now we know, I've told you that we know a lot about and are able to identify reliably the species of the fungus, and we have some idea about the diversity of the cyanobacteria, and we can keep track of units. Let's now move into the third thing that I promised you, which is how are lichens helping us understand the web of life better? So you can start looking at that question by saying, okay, what does this network or this web between them look like? And remember, instead of showing you the actual web, which looks pretty pretty but has not much information, I'm going to be showing you matrices which show the interaction. So if we look at the Peltigerous species and the nostoc in their interactions globally, we find something like this. So this is the same that I showed you earlier, but just with many more species. On the rows, we have different nostocs, and in the columns, we have different Peltigerous species. And whenever there is a box in here, it means that there is an interaction. So what you can see is that there are, again, these boxes that have a lot of interactions within them that are indicating that there are certain groups of species that are interacting with each other more than they are interacting with the rest. So these modules. And so we call this a modular web. Okay, so that's the structure when you look at it at the Peltiger and the nostoc from the entire world that we have sampled. And so it turns out that another thing that we're learning from this is that actually the species that are in each one of these modules, they're not random. Oftentimes, the species that are part of the modules are closely related to one another. So to be able, this is one of the reasons we needed those genealogies to be able to uh, tell these kinds of insights. Now, that was kind of like at a global scale. But I'm interested in looking at what happens when you look at that structure at different spatial scales. And so I'm in a collaboration with Dr. Diane Hoagland, which is a lichenologist at the Alberta Biodiversity Monitoring Institute. And they're doing something really cool. So this is a map of the province of Alberta in Canada. And they basically, this institute turned it into a grid. And every 20 kilometers, they are doing surveys of biodiversity to inform conservation management and to study biodiversity in Canada. And so they collect a bunch of things, including lichens, and they have over 8,000 Peltigerous specimens in their collections. And so through the collaboration that I have with them, I've been able to get my hands on these specimens, and I'm basically sequencing them, looking which nostoc is interacting with which Peltigera where. And so I'm able to describe these webs at multiple scales. This sampling spans different areas from the Rocky Mountains to the boreal forest to the prairies. So we have all of this information about that allows us to ask questions about how these interactions are changing in different places. And so what I'm doing is essentially I have a subset of those sites, and I got my hands on the specimens that were collected from those sites. There's over 3,600 of them, spanning more than 30 species of Peltiger and more than 20 different kinds of nostoc. So by knowing who's interacting with whom where, I can ask how are these interactions changing across scales and gradients. So to give you some idea of what we're finding with a subset of the data that we have gotten so far, let's look at what the web of interaction between Peltiger and nostoc looks like, but now instead of at the global scale, we're looking just in the province of Alberta. So this is again a matrix of interactions. Rows are different nostocs, columns are different Peltigeras, and whenever there's a box, it means that we have an interaction. Now, if you remember, what I showed you earlier is that we had these modules, right? These groups of species interacting more with one another. But here, we don't have those modules anymore, and it's the same system. It's only that we're now looking at it at a different scale. What we have here is that there are some nostoc here on the top that are able to interact with many Peltiger species. And then as you go down in the matrix, you start seeing nostoc that are able to interact with less and less. And then as you go down, those that interact with less are interacting with a subset of the most generalist one. So this is a structure that we call nested. So it's the most specialized ones, the partners are nested within the most generalist. And it's different from what we see at the global scale. So one of the things that this is teaching us about the web of life is that depending at which scale you look at these webs, their structure changes. And understanding why that happens, again, since it has consequences to our understanding of whether or not species are going to persist, is very important. So this is kind of what we're doing now. But that's one insight that is coming out of this, that the structure is changing across scales. Now, if we take a more detailed look, we focus here on these maps are showing the distribution of different types of nostoc across the province of Alberta. And so you see, for example, that these pink nostocs, they are all over here in this northern part. They are the most dominant over there. But then as you move here to this darker part, which is actually showing the Rocky Mountains, you start seeing no longer seeing the pink ones, and then there's the red ones. So it means that this web is not homogeneous. As you move from one place to another, the players in this web are changing. And then another thing that we're finding is that some species are able to associate with some partners. For example, some Peltiger species are with the pink nostoc when they are up here, and then they are with the red nostoc when they are here in the mountains. So some of them are able to switch the nostoc that they interact with depending on where they are. But not all of them can do that. Some of them can only interact with blue, and therefore you only found them here and not here. And now that's a very important insight. It means that some species are able to switch their partners depending on the conditions, and some species are not. And that gives us an opportunity to study why some are able to do it and why some aren't. Because if you think about managing biodiversity, if all species were able to switch, then it doesn't matter much if you lose some part of the land. They're all going to be able to persist because they can interact with this other partner over there. But the situation that we have here, what this is teaching us, that some are able to do that and some don't. And so some are going to be more vulnerable to that change if there are changes in the landscape, because they're not going to be able to interact with other things. And so these are just some of the insights that are coming from studying lichens. The structure of these webs has information about whether the species are going to persist or not. When we study them in lichens, if you look at them at different scales, they have a different structure, so their dynamics are different. And then if you start looking at the details, you start finding things like this, like it's not homogeneous. Some are dominant in one place, but not in others. Some species are able to switch, but not all are. And so the question is then, why are all these things happening? So I want to end with a note, because I've been, of course, highlighting a lot of my own work and the work that's been done from my lab, but some of these motivations to start to study these interactions in lichens date back to a while back to the early 2000s. And then recently, there's been a surge of many different lichenologists are studying these interactions in different groups of lichens, finding completely different patterns to what we see in Peltigera, which again, it's cool because then it gives us the opportunity to say, okay, when is this pattern emerging? When and why not this other one? And so I'm just here kind of highlighting different studies all within the last couple of years that are looking at that in different groups. And it's a very exciting time to be studying these patterns. And I just put here the ones that fit in the slide. There are many others. But essentially, this scheme that I put here recently made it to the cover of a very prestigious journal. So it's just an indication that we really are learning um, a lot about this web of life by studying the interactions between the symbionts in the lichens. So yeah, I hope that I delivered on the three messages that I promised you. That lichens are the symbiosis. You have a fungus living together with a photosynthetic organism that can be a green algae or a cyanobacteria. I hope you remember some of the cool facts. Wet them if you want to see the really cool colors. Um, that the web of life is this idea that biodiversity is a collection of interactions, and how those interactions are structured has information of as to how the community or whether or not communities are going to persist in time. And so we want to understand how they're structured, when they are, how in different ways, what shapes them. and then and then that lichens by this their nature are teaching us a lot about how the structure is different if you look at different scales, how there's variation in the ability of species to switch partners or not, and things like this. And so with that, I'd like to thank the many people who have uh, contributed to some of the work that I presented to you today. People in my lab, collaborators, my partner, my boy Moises, who's done a lot of the vector art that I showed you during the presentation. And thank you, and thanks to the Torrey Botanical Society for the invitation. And I'll be happy to take questions now.
Thank you very much, Carlos. That was a great talk. So while everybody, again, as I said before, feel free to write your questions in the chat here, and then I will read them out to Carlos for him to answer. Yeah, the art and the pictures, they were fantastic, and awesome figures too, like really high quality. It's great. Thank you for joining us to give this presentation.
It was my pleasure.
So while people are sort of thinking about their questions and putting them up here, I have a couple. I have at least one.
Yeah.
um, so your observations that you were seeing that some Peltigera, they varied in their photobiont depending on like where they were in Alberta. So there's like a scaling or a geographic element to this.
Right.
Um, so I was curious if you were observing, or if you could speculate on whether or not this was due to, you know, more of a like a population genetic variation kind of thing, where like there's some level of structure in the population in that area, and so they're like seeing different strains of nostoc, or if it's more along the lines of like a niche optimization for the specific habitat that you find the Peltigera in.
Right. Yeah, that's a great question. And since we have genetic data, I do have some insights about it. So, for example, for the case where I was saying that I was talking about some of the species of Peltigera that when you find them in the boreal forest in Canada, they are with the pink nostoc. And when you find them in the mountains, they are with the red nostoc. It turns out that, yeah, they are the same species of Peltigera, but there is some population structure. So it looks like um, there's like a, yeah, like a subpopulation that is in the mountains and they are mating more amongst themselves, and then another subpopulation that is in the boreal forest. So as we so that's from from a little bit of genetic data that we have, which is like short ITS sequences. But we're gonna be adding genomes to this as another layer. So we're gonna be able to know in a more fine detail if maybe we are seeing early stages of speciation in some cases that is driven by finding a new host. There are other studies in different parts of Canada that have shown that um, even if a different situation where even if the nostoc is in different places along a gradient, sometimes the Peltigerous species is only in a part of that gradient because it cannot potentially tolerate, a different temperature or a different kind of precipitation. So you have both things. I have observed the population structure kind of thing, and the niche difference kind of thing has been shown in other studies. So
Very
yeah,
cool. And I'm sure that they play into one another. You know, it's sort of
yeah.
like which comes first. One follows the other sometimes. But that's awesome. And it's also really cool to hear that you're starting to get like, you know, genome data for the Peltidra and also the nostoc.
Yes, correct.
That's very exciting. I'm very
Yeah.
excited about that. So I'm going to start reading some questions. So Amy B says, "Great talk. So of the lichens that you've studied, which do you consider the most vulnerable?"
So there are, I don't have a picture of them here, but there's this illustration of this species that's called Peltigera soradians, which is mostly found in the prairie um, region of Alberta. And we've always found it um, very tightly associated with the same nostoc that is not one of the most common ones. And so the prairie in Alberta is one of the most threatened also kind of biomes. So those species like that, like these Soridians that are seem to be very specialized on nostoc that are not super common. And on top of that, they are often in environments that are threatened by a lot of anthropogenic pressures are the ones that seem to be most vulnerable. But I want to emphasize here that the data that I've shown you is a fraction, like a 10%. So at the end of the project, I've sequenced like 200 something and we'll get 3,000. So we still have long ways in terms of data accumulation. Maybe some of that story is going to change as we accumulate data, but so far that's kind of what it looks like.
Okay. And I know that it's fairly well known that a lot of cyanobacteria, I'm sorry, cyanolichens tend to be very threatened by elements of pollution. I'm not really sure what that kind of looks like in Alberta. Um, is that much of a threat there, or is it more like habitat destruction?
Yeah, that's a good question. So what I know about indeed, I think Peltidra and cyanolichens in general, is true that they are, among the on the more sensitive side to pollution. And so I can speak to a like anecdotal pattern where around urban centers in Alberta, there's more deposition from pollution just by virtue of being closer to urbanized things. And then the richness and the abundance and the sites that are around urban centers is much lower than when they are far away. Um, so I suspect that the position of pollution could be one factor, but there might be other things. So this is kind of like anecdotal what I know about it
Yes.
so far.
Okay. We have a question from John Franklin. He says, "Is there any noticeable physical difference in the Peltigera species when found with different nostoc varieties, or do they still look the same?"
Right, that's a great question. In my experience, most of them don't seem to have any noticeable difference, but there are some. I had a picture. There's one particular group where I have seen it, which is this one right here in the middle, which is a picture of Peltigera Malaysia or Malasia. Um, and there are different, I have seen thalli that have different shades of that blue, which is, by the way, very, very pretty when they are with different nostocs. So I have seen it, but it's not the norm in the species. But at least some of them seem to have a change in the color.
It's very nice to think about that just minor strain differences in just the nostoc they associate with can change their appearance like that. Um, I know that there's like I think that at past botany meeting there was a lichen talk that was really exciting. That was like these ones believed to be two different species of lichens were in fact revealed to be one tripartite lichen that had both um, the
Yeah,
cyanobacterium and also a green alga, and it just looked so radically different.
yeah,
So
yeah. For the symbiotes, yeah, it's very interesting. I also just came to my mind. I think one of my collaborators, Nick Maga, has in the group that he studies. I think he has also seen differences in the color of the nostoc of some species. But I think it's definitely not the rule, but it happens sometimes.
okay. I have a question from Sarah. Sarah says, when you collect lichen specimens, how do you make sure not to disturb the organism slash colony too much, as well as keep the collected specimen alive outside of its environment?
Okay, so there's two parts. The keeping it alive, it depends on your purpose. Usually, when I collect lichens, they are meant to be herbarium specimens, so we just dry them, and we don't care about them staying alive, even though sometimes they can, you know, potentially be alive if you rewater them after some time. But for the most part, I do not intend for them to stay alive when I collect them. There's some people that do certain experiments um, where they need to collect the lichen, bring it to the lab, and keep it alive. And so what they do then is subject the lichen to these cycles of wetting and drying. So they basically water it, let it dry, and then after a certain number of hours, they water it again to kind of try to mimic that fluctuation that they have in nature. And so that way, multiple people have been able to, keep them metabolically active after they've been collected. And then as to not to disturb them, so we usually try not to take the whole specimen. So the Peltidra in particular, it has this morphology that is kind of leafy. So we call each one of these um, parts, we call them a lobe. So when we collect, we try to take a representative piece that is big enough for us to see the morphology and get DNA samples. And that's usually, a few lobes, but you don't need to take the whole thing for that. So that's kind of how we ensure that we leave some of them because we take a few lobes and that's enough for what we need.
Yeah, and I'm just going to also just say, like, for anybody thinking about like taking a lichen from the environment and trying to grow it at home, I'm going to recommend that you don't do that. Generally, because a lot of lichens, especially these cyanolichens, are threatened in various ways, and also they're fairly notoriously difficult to grow at home anyway. And so it's a challenge is probably going to be unsuccessful anyway.
Yeah, thanks for adding that. I should emphasize that the people that have done this is for particular experimental purposes, but
And
not
usually under
for
very specific laboratory conditions.
exactly. Yeah.
Um, yeah. So So Amy B also asks how to access this recording, and I have to figure out how we're going to actually put this up. It might go up on maybe a dedicated YouTube channel. It might go up on the Torrey website, but we will make this recording available. And when I figure out how best to distribute it, I'm going to make a post on the Facebook page, and and maybe make sure that notification goes out to Torrey members that it's up, just so that people who were not able to attend today are able to listen to this really cool presentation. Um, Zihao says, to observe and identify these lichens, what kind of magnification is needed?
So usually lichenologists have this hand lens that have 10x magnification is pretty good to see most of the structure. So usually these 10x lenses are sold, are catered to people who do jewelry stuff. So that's how you can find them. So 10x is usually a good spot.
Okay, I think that this pretty much wraps up the question time. We spent a few good minutes talking about this stuff, so I think we're going to let our speaker go. A lot of people are saying thank you very much for the great talk, Carlos. This was really exciting.
Thank you. Thanks to all of you. I saw just quickly a question about whether there are studies like this in the chlorolichens, and I just want to say yes,
Ah,
and there's many coming. So a few of the ones that I highlighted at the end are actually on the green alga. But yeah, so thanks Jordan, and thanks everyone who stayed and asked questions. I had a lot of fun preparing, and I hope you got something out of it.
I hope so too. So thank you everybody for stopping by and enjoying this talk with us. Our next talk is scheduled for December 1st. I know that's coming up quite soon, but we're going to resume our usual schedule. This one was a little bit different because of Carlos's schedule and also because of election day. So we didn't want to overlap with any of that. Um, but our next talk is going to be pretty exciting. Still waiting on getting the final write up, the abstract for the talk from our speaker. But it's going to be pretty cool. It's going to be sort of about this intersection between botany and species identification and modern technology and measuring tools and things like that. I think it's going to be really cool. So I hope that that people will join us for our next talk, December 1st. Again, notifications will go out on the Facebook page and also directly via email to Torrey members. So please stay tuned for an announcement, and I hope to see you there. So again, thank you everybody. Thank you, Carlos. Very much appreciate it. I look forward to talking with you again soon.
Yes, my pleasure. Let's stay in touch. Bye,
Definitely.
everybody. Thank
Goodbye.
you.